Thank you.
Yes, so I'll just welcome everybody again. Thank you for joining us for this webinar series. Today will be a first session where we will talk about introduction to next generation sequencing and focusing on Illumina technology and principles behind it. Illumina's mission out there is to improve human health by unlocking the power of genome. And there are many aspects of human health that we can focus on. Just to name the few, we can look at reproductive health, where we can look at non-invasive prenatal testing to screen for chromosomal trisomies and avoid more invasive procedures like amniocentesis. We can also look into oncology applications, where we can look for certain predispositions in family members for certain cancers, but we also can look into certain mutations and pathways that can help with personal treatment, such as personal medicine, but also to monitor for possible relapse. You're all aware that currently we are in a huge pandemic, and NGS was used to identify this novel coronavirus that we are all dealing with, and we are currently constantly monitoring the situation, surveilling the spread of the disease, and also looking at mutations that follow with this virus, but also possible co-infections that might be present. Also looking into developing the disease and looking into developing the vaccine and looking at the vaccine trials. And in all of these aspects of infectious diseases, NGS is very present. You will remember that 2-3 years ago we had a huge listeria outbreak, and NGS was also used in this instance to pinpoint the source of this spread of infection or disease. We can also not directly look at improving human health, but also indirectly at improving our food through agriculture, looking at specific plant and animal species as our food, and we can look at that market-assisted breeding to increase the maybe quality of milk or meat, or to look at improving the yield of certain cereals, for example, or to look at the drought resistance and improving those traits. So there are many different aspects out there that we can look at when it comes to improving human health. Initially, Illumina was more research orientated, and many of these instruments and technology were present at universities and academic researchers and the big genome centers. But now these days they are moving more towards more clinical aspects and more into hospitals, precision medicine, and pharmaceutical companies. And this wide uptake of Illumina technology is based on the high quality of data and also low error rate. And you can see on this slide that there is a huge install base out there in the world globally, with more than 13,000 different active instruments. And I'm really happy that South Africa and Africa as a whole is also on this map, and that we are also doing the NGS very, very actively. So why is there such uptake on this technology? It's because of the simplicity of using the instruments, but also for having that integrated end-to-end solution where we have sample to answer workflow, where we have we can start with our library preparation. We have solutions to sequence, but there are also solutions how to analyze the data, and to some extent maybe through the third parties how to interpret that data as well. So obviously sequencing is not new to us. We've been doing this for over 40 years, since the Sanger and the colleagues invented this technology in 1977, and they sequenced the first bacteriophage, which was a 5X174. So we've been doing this for a very long time, but where the boom in the technology is that, and with the Illumina sequencing, that we are able to all of a sudden do massive parallel sequencing, where we can do thousands, millions, billions of data reads at the same time. And this has really opened up the space to all these applications that we spoke about just now. So just briefly to refresh your memory on Sanger sequencing, because that's how everything started. this is a technology that uses deoxynucleotides that are labeled, which are chain terminating nucleotides, and these different lengths reads are then separated on a capillary gel, and the laser will excite these labeled nucleotides, and the computer will read off the sequence. So we will know, we are able to determine the order of nucleotides in that specific fragment. And this is still a gold standard when it comes to sequencing, and it's a really good technology, and we use it a lot. But of course, it has its limitations, and it's also, when we look at the large projects like de novo sequencing or gene expression, or when we are looking at a very low frequency variant sequencing for somatic applications, it becomes a cumbersome process and not a cost-effective process. So, and you're also very familiar that we can only do about 600 to 800 base pairs at a time, and we can only do it in bidirectional, so we can only do reverse and forward. And this is, and this becomes very difficult when we are looking at the heterozygous calls, because we have loss of signal or signal overlap. When we look at the, sorry, yes?
Sorry, I just want to, on behalf of some of the participants, ask that we switch off our video. They say it's slowing down audio quality for some of the participants. Yeah. So if you can please just turn off the videos in the meantime. Thank you. Sorry, Maria, you can carry on.
No problem, yes. So yeah, just please keep everybody muted and just switch off the videos so that we also have a clear image on the screen. So as I was saying, with Sanger sequencing, we can only do a bidirectional workflow, only forward and reverse, as you know it. But with next generation sequencing, we can do this massively parallel sequencing. Sorry, Adrian, just mute everybody, please. So we can have thousands of reads of the same on the same locus, where we can look at the overlapping reads, align them together, and then have a quantitative and high confidence variant calling. And this kind of produces kind of digital data where we can analyze it independently and quantitative. So this kind of technology really has huge applications, like we saw in cancer genetics, in rare human genetic diseases, in agriculture, microbiology, reproductive health, and so on, because it allows us to look at the whole genome sequencing, also possibly at the whole exome sequencing, where we can look at the single mutations, or we can look at the copy number variations, structural variations, but we also can look at the RNA sequencing, where we can look at the differential expression, different alternative splicings and gene fusions, but we also can look at the epigenetics and the modes of change, and how those modifications are influencing gene expression. So when we started sequencing, there was a huge initiative to do a human genome project in 1990, and that's when it started. And of course, we did this. It was a huge international project, and it was done on the Sanger sequencing. But of course, it took more than 10 years to get to the human genome, and also it costed $3 billion, which is a huge amount of money. With the boom of technology and having it more readily available to people, and also being cheaper, we see this boom of cost per genome being lower and lower every year. But also, obviously, because it's now more affordable, we have millions of genome sequence out there. And even though you saw on the previous slides that we did 100 petabytes of data in just 2018, and this is a number with 15 zeros, just to put it in a perspective for you, and that was done just in 1 year. We still haven't sequenced, we only sequenced 0.01% of species out there, and only 0.02% of human genomes have been sequenced. And just to put that in perspective, that's about 1.5 million of humans being sequenced. And even though we generated all of this data, we are still trying to decipher 99% of variants in human genome alone, yet alone in all other genomes and species that are present on Earth. So when it comes to other technologies that we commonly use in the lab and that have been in the lab for a very long time, I just wanted to kind of show where NGS fits and how does it to some instances complement the qPCR Sanger sequencing that we are doing. Those are both great technologies and they have their place in the labs, but they lack complexity, they lack the discovery power. So we cannot do multiple targets at the same time. We can only do a few, depending on, let's say, qPCR instrument and how many channels and different fluorophores we can detect. We can do 4 or 5 targets for one sample. And also we have to have a prior knowledge of the sequence that we are working with. So we need kind of NGS in order to decipher that novel species, let's say, or novel targets that we want to look at, and then we can create a qPCR assay that can help us with, for example, like we did with COVID testing, where we had NGS help us identify the new species of the coronavirus, and then we developed qPCR assays for a fast screening of positive or negative samples. And if you have to kind of upscale qPCR and Sanger sequencing, of course, more samples, you want more targets, you want to have, of course, the price of it increases. With NGS, the cost of that sequencing reagent, so the sequencing kit, is fixed price, and more samples you put in there and more targets you put in there, it actually becomes cheaper per sample and per amplicon that you want to sequence. So NGS opens a door to massively sequence multiple samples, multiple targets, so it becomes really a cost-effective way of doing it. So like I said, it was a prelude to why is the NGS solution, is because we are able to do simultaneous hundreds of targets, many different gene regions, many different, looking for many different variants, but we also able to, because we are sharing that cost of a sequencing kit amongst many different samples, we are able to downscale the price, but we also just need maybe one DNA extraction where we can look at all these multiple regions, and we can conserve our precious sample, by having a very low input of DNA. And of course, with NGS, because we are sequencing so much more, so much deeper, we have that sensitivity and resolution of data, and we can zoom in so much more, especially when it comes to let's say, a cancer research, where we can look at the limit of detection with it. So it really becomes we are able to enhance this discovery of genetic variation with NGS and look at multiple mutations such as SNPs, insertions, deletions, duplications, inversions, and so on. But we are also able to look at disease resistance, genetic risk factors, and morphological differences, and also use this technology to improve on certain breeds or genetic modifications, through genetic engineering or synthetic biology or cloning. And also we are able to understand the genetic drivers of the disease, because we can now zoom in more into copy number variation, gene expression, gene regulations. We are able to understand so much more when it comes to human health. And for me, where kind of NGS created a big difference for when it comes to amount of data that we are working with is obviously microbiology and infectious diseases, because we can discover new species with such ease. We don't have to have any prior knowledge of the bacteria or virus that we are working with, and we can also look at the co-infections and quasi-species out there, and we can also be able to track different mutations and characterize those pathogens, and also look at the multiple samples at the same time, but also look at the pathogen and host interaction and see how is the host, what are the kind of genes switched on or off during that infection. Also Another big field where NGS made a huge difference is oncology and human genetics, where again we don't have to have any prior knowledge of the variants that we want to look at. We can look at the multiple variants at the same time, which obviously decreases the turnaround time that with which we can provide clinicians with a certain recommendations for the personalized treatment. So it becomes really a higher turnaround time, but also we can zoom so much more into the somatic variants. Like I said, we can do the limit of detection for NGS is 3 to 5%. So this really makes it a huge difference. So this slide just represents many different applications that I spoke about just now. Of course, there are many more, and this is just to illustrate that there's many. Of course, there are many questions out there, many research, that we can do. So regardless of what is your biological question or applications that you want to follow, the workflow of the Illumina NGS will be more or less similar, and it will always be, it will always start with the nucleic acid extraction, because you need to work either with DNA if you want to look at the whole genome sequencing or exon sequencing, or you need to extract RNA if you want to look at let's say a differential expression. And this is just a very general workflow, but I would say it's true for all samples out there. You will start with of course nucleic acid extraction. We will have to QC it. We'll do then. There are many different types of library prep, but and we will go through some of them, followed by library QC and then sequencing on many different instruments, and then of course data analysis. So what we wanted to do with this NGS webinar series is to break down this workflow for you, and we decided to spend 2 weeks on nucleic acid extraction and QC. Because this is the first step, but also the most important step. Remember that there's that saying, junk in, junk out. So if your nucleic acid, if your DNA or RNA is very bad quality, your library prep will also not be, not have a good quality. But also if your library quality is not that great, or you have very short inserts or something, then your sequencing data will also not have a good quality, and you will see that obviously in your analysis. And it's very expensive workflow to find out at the end of it that the data that you have is not optimal, and that you maybe not even use it. So it's really important that we have these QCs in place before we come to the end of experiment. Okay, so we're going to have a series, we're going to have a webinar next week where we're going to concentrate on nucleic acid extraction, and that will be particularly for environmental samples, but also for plant samples, and also from a more clinical point of view for blood and tissue samples. And we will also look at certain QCs that need to be followed. But just to mention here briefly, when it comes to nucleic extraction and what is kind of needed to go into a library prep, because every library prep kit will have its startup point that it needs. So obviously that will be a specific concentration, but not everything is in the concentration. We also have to have a good integrity and the purity of that nucleic acid. So when we talk about concentration, when it comes to NGS workflow, we usually measure this with some kind of fluorometer, either qubit or picogreen, but we also want to assess the purity of that sample to make sure that there is no inhibitors in it. For example, those of you that work with soil, that it's very difficult sample type, and that it has many different inhibitors in there, and that downstream analysis can be tricky when it comes to that. That's why we decided also to spend a whole session on just talking to you about extractions and how to improve on that. And also when it comes to nucleic acid, we should look at the integrity of it. Of course, you don't want your samples degraded. There are some samples that we cannot get away with that. For example, FFPE samples, but majority of samples out there, of course, we don't want degraded DNA, because that will create very short libraries, which in turn can diminish your sequencing data quality. And this is more true when it comes to RNA work. You know that RNA is much more easily degradable than DNA. It's a little bit more trickier to work with. So it's really important to do RNA QC and look at it through some kind of fragment analyzer, like a bioanalyzer or tape station or a lab chip, where you can assess the integrity of your nucleic acid. Okay, so please join us next week when we discuss this in nucleic acid extractions in more detail. We will also have we will spend 2 weeks on sequencing and on different instruments and applications behind it. So we will zoom into 4 different instruments and how and what applications you can do on them. And then we will also have a separate session just for data analysis and to look what is possible from Illumina's point of view. Okay, today I will talk a little bit more about library prep more in general, the principles behind it, and also the principles behind the Illumina sequencing technology. So the NGS workflow, like you saw, it would start obviously with DNA or RNA extraction, and then we do library prep followed by cluster generation and sequencing, which actually happens on the NGS instrument, and then the data analysis. And regardless, like you saw, regardless if we are working on DNA or RNA, the principles behind library prep is similar. And also depend, it doesn't really matter. Are you working with small organisms like viruses, bacteria, fungi, or you working with larger eukaryotes, like different agricultural important species of plants and animals, or if you're looking, if you're working purely with human samples. DNA is DNA, and the way we prepare the libraries, you will see just now, will follow the same principle. Okay, same with RNA. If you want to look at the whole transcriptome work or alternative splicing or fusions of genes, there are many different library preps out there. This slide just illustrates very briefly how libraries are constructed. So with RNA workflow, obviously we need to convert that RNA. We need to do a reverse transcription to cDNA, which will then follow. We will ligate what we call adapters onto our DNA. You will see on the DNA, first we need to fragment it, and then we will ligate these adapters, and you will see why these adapters are so important when it comes to library preparation. Like I said, with the DNA library prep, we have mechanical fragmentation, which can be done with the ultrasonicator such as Covaris, and these kits are a TruSeq series of kits, namely TruSeq NanoDNA and TruSeq PCR-free, but also Illumina has an enzymatic fragmentation kits such as Nextera Flex or Nextera XT. So very briefly, when it comes to TruSeq kits, let's call them that way. They are called that way. We will have genomic DNA fragmentation with Covaris, so we are mechanically shearing the DNA with Covaris. Then we will follow by end repairing that, so making sure that our DNA is blunt-ended. Then we will do a narrow size selection, and then we will adenylate those fragments in order to have an A on the end of the molecule, because our adapter, you can see it here, it has a T on the other side, and you know that A and T are binding together. So when we do the ligation of the adapter, that T will bind to the A and we will have a molecule. Okay, this is the same principle that happens with RNA library prep, except at the beginning we will have that RT conversion of RNA to cDNA, which would be a first strand and second strand synthesis that we get to the cDNA, and then we will start, let's say, from point D, where we'll adenylate and then ligate the adapters to it. Okay. Enzymatic fragmentation workflow works slightly differently. This is an Nextera XT workflow. So what Illumina has done is they've taken a modified transposon, which at the same time can share your DNA, so you can cut the DNA, but can also insert this, let's call them blue and green tags. So you can insert these tags into your DNA, which will then serve as a landing site to our adapters. Yes, sorry.
Sorry, it's Rendice. I just, I'm not sure if the correct slide is showing on our side. It's still on the mechanical fragmentation workflow slide.
I think it's just low. Are you seeing Enzymatic now?
Nope.
I can see
Maria, I can also see Enzymatic. I think
So
it's
maybe
from
some
your side.
people's internet is just slow. Okay, that's fine.
I'm seeing Enzymatic as well.
Okay, thank you. I can
Okay,
hear you,
remember-
so it's fine.
Okay. Just remember, we're also recording, so it will be available to you as well. Yeah. Okay. So as I was saying, we will have then inserts in our DNA to which the adapter can bind to and then create a library. Nextera Flex, which is a newer version of Nextera kit, has this transposon bound to the bead. So it's much quicker workflow, but it also it makes sure that we oversaturate that bead, so we don't have overfragmentation of our DNA. Okay, and it also it gives us a good size distribution when it comes to library prep. So what are these adapters that I keep talking on? And here I'm illustrating. You have your DNA, and in the end, it doesn't matter what kind of do you follow, a TrueSeq library prep or Nextera library prep. Your DNA molecule at the end will look the same. Okay, simple is a simple representation. So you will have your DNA, your insert, for which we don't have to have any prior knowledge, and on each side we will have what we call the adapters, and these adapters are about 60 to 65 base pairs, and those adapters have, you see here, three distinguishing sections that are really important when it comes to NGS. So the first section will be what we call a P5 and P7 region, which actually allows our molecule to bind to the flow cell. Okay, and you will see later on what is the flow cell and how do we actually sequence. But this allows us to bind that molecule to the flow cell on which we will do the sequencing. Then we have what we call an index or a barcode, which is a unique sequence that will distinguish samples. So remember, with NGS, we can multiplex many different samples in one run. So we can put hundreds of samples in one run, and these sequences then distinguish those multiple mixed fragments that are now sequenced all as one together. Later on, we can distinguish between those fragments bioinformatically and pull them out and say to which sample they belong to. Okay, and the third region that we have on this adapter is a region to which the sequencing primer will bind. So once the sequencing primer binds to it, you will have the extension of your DNA insert. And because we have this region on our adapter, this is the reason why we don't have to have any prior knowledge of that organism that we are sequencing. So we can do de novo sequencing because the primer will bind to this specific region regardless of what our DNA insert is, which obviously for genome sequencing will be multiple different inserts. So this is just to illustrate many different library types that are available out there, obviously for many different applications. You can always reach out to us and discuss this with us, but also Illumina has this library prep Selector webpage where you can actually search by area, application or method, and it will list for you many different library preps that are available out there for your specific needs. Okay, when it comes to Illumina sequencers, there are many sequencers out there, and it's important to mention that regardless which library prep you use, all of those libraries, obviously if they're for Illumina sequencing, can be sequenced on many different platforms, and you can sequence that same library across many different platforms. This is just to illustrate that Illumina has instruments for every needs, where we're talking about very small genomes like your viruses and bacteria, where we can also work with small genomes, but also slightly larger ones, eukaryotic genomes, and also humans, and also where we can look at the population and start sequencing populations. And these are really huge instruments with very high output. As many of you are aware, there's service providers out there that have many different instruments. MySeq is obviously the most popular instrument in the world, but also in South Africa. So most of these service providers will have a MySeq as a smaller output instrument, but they will also then have a larger platform like HiSeq 2500 or a NextSeq 550. Okay. This is just to illustrate how, we have smaller instruments, and obviously the capital, the initial expenditure is short, but we are able to, is small, and we are able to procure it, let's say, rather easily. But obviously the output of those instruments, it's low, but as we are moving up this scale and we're looking at the production scale sequences, we're looking obviously as a much more capital expenditure, but the sequencing per gig becomes much more cheaper, and we can sequence many more samples on it. So if we are looking, for example, at the iSeq, let's call it the baby of the instruments. It can do only 1.2 gigs of data, and to put it into perspective, when we are looking, let's say, at the bacterial sequencing, you can probably, at the 100x coverage, you can probably sequence 3 samples. When it comes to a MySeq, because it has a slightly higher output, you can maybe sequence 30 genomes. But when we look at the NextSeq, for example, we can do as many as 300 bacterial genomes on it. Same with, let's say, human genomes. When we look at the whole exome sequencing with the NextSeq, with the NextSeq 550, we can do 12 whole exome sequences. With the latest addition to Illumina portfolio, NextSeq 2000, we can look at 40 different samples for whole exome sequencing. And with the NovaSeq, which I like to call a T-Rex of Illumina portfolio, we can do up to 500 whole exome sequences. So just to give you an example of what is possible out there. Like I said before, we're going to have 2 sessions on specifically benchtop instruments, and we will look into each instrument, so into iSeq, MiniSeq, MySeq, and NextSeq specifically from technological point of view and the outputs it can do, and specifically what applications can be run on each instrument. So please do join us on the 31st of August, where we will be discussing iSeq and MiniSeq, and on the 7th of August, where we will be discussing the MySeq and the NextSeq. So this is just to illustrate what happens on the sequencer itself. So once we have a library, we can dilute that library to picomoles, and we can denature it, and we are ready to load. This side here on the right hand side is the cartridge and the instrument. This is an NextSeq 550 to the scale, so you can see how massive the reagents for the sequencing are. We will have a buffer. We have something called the buffer, which will obviously have an incorporation buffer in there. We will have what we call a reagent cartridge, which will have many different reagents in there, your nucleotides, your cleavage, your all the reagents needed out there for SBS sequencing by synthesis technology. And we will also have a flow cell to which our library will bind, and the whole sequencing will happen. I just wanted to illustrate for you many different flow cells that are available out there. Obviously, each flow cell is specific for a specific instrument, and so are the reagent kits. So this is specifically for a NextSeq 550. NextSeq 2000 flow cell looks like that. This would be a NovaSeq flow cell. We have a MySeq flow cell, and then the iSeq flow cell as well. I just wanted to illustrate to you how they look like. But regardless on what instrument we are doing the sequencing, and that will depend on your application and the output of data that you need out there. The principle of Illumina NGS sequencing is the same. So we will have something what we call a cluster generation, which obviously happens on the flow cell, and then we will have sequencing by synthesis, which means that as we are synthesizing the complementary strand of our template, the label fluorophores will give off the signal, and the camera will pick up those signals, and it will record the order of those nucleotides. Okay, so let's zoom into cluster generation and how does that happen. So first of all, a cluster is clonally amplified target region. So our library, the first step is to clonally amplify our library and to create thousands of copies of the same molecule. This is just to give enough signal to the instrument to detect these clusters. Just to zoom in onto the flow cell, you saw there is many different flow cells out there. This is a high-seq flow cells, but basically the principle behind the flow cell is that it's a thick two pieces of glass slide, and in between those two glass slides we have a loan of oligo that are complementary to adapters that are sitting on our library. So these oligos here that are on the flow cell are complementary to the adapter portion, that P5, P7 portion that is sitting on our library. Okay, and there are, just to mention that there are two types of flow cells, a random, what we call a random flow cell, which means that this pool of oligos are randomly spaced on the flow cell, and then we have what we call a patented flow cell, which is we have micro wells on those oligos. On that flow cell, and we have oligos in the wells itself, and there is no oligos in between those wells. So we can, and those micro wells have very defined size and spacing, so we can with ease increase the cluster density and oversimplify the imaging, which makes it much more faster to sequence as well. So I will just go briefly into what happens on that flow cell and how do we get to cluster generation and sequencing. So the first step obviously is when you load your samples into a cartridge, it will flush it onto the flow cell, and your library will bind to the oligo on the flow cell. It will then do a 3' extension to create identical or complementary strand, and the original will then be washed away. As soon, because we have the oligos, those adapters are kind of attracted to the oligos that are sitting on the flow cell, and they naturally flip over and tend to create what we call a bridge. Okay, and then again we have polymerase which will do the extension and will create a complementary strand, and this happens across the whole flow cell simultaneously for thousands, billions of reads at the same time. Okay, so and once we create these bridge amplification molecules, we are ready to sequence, and obviously we'll linearize all those bridges, and we will block the 3' end so that we don't have that flip over happening again. And then with the introduction of a sequencing primer, the sequencing which will bind to the specific region on our adapter, we will have sequencing. So and this happens one nucleotide at a time, where we have a labeled nucleotides which will then bind to the corresponding nucleotide on the template, and it will give off the signal and the camera instrument will record this. This is the basic principle behind SBS technology. Different instruments out there have what we call a 4-channel chemistry, 2-channel chemistry, or 1-channel chemistry. So your MiSeq and a HiSeq will have what we call a 4-channel chemistry, where each nucleotide has a different label, and we are taking 4 different images. Your NextSeq and MiniSeq will have two different labels. We will take 2 different images, so our C will be red, our T will be green, and the A will be mix of that. And the latest technology out there is 1-channel chemistry, and this is for iSeq 100 only, where we'll have intermediate chemistry step between the two images. So T is always labeled and G is never labeled, and we will take first image, then we will have intermediate step where a dye will be cleaved off from A and it will be incorporated into C, and then we look in between those 2 images and we will call the base call. So if we have if we don't have any images, if we don't have any signal in both images, we will call that a G. If we have a signal in both images, we will call it a T. If we have a signal in first image, we will call it A. If we have only a signal in the second image, we will call it a C. We will go into a little bit more detail on this when we discuss each instrument. Okay, and as you know, Illumina sequencing is also a paired end sequencing. So what that means is that same molecule, once we are done with what we call a read 1, we flip that molecule around and we start sequencing it from the other end. So it's not necessarily forward and reverse like we know it with Sanger sequencing. It's the same molecule. We're just flipping it. We're doing a turnaround, turn over, and we are sequencing it from the other side. So if it was sequenced from P7 region, when we flip it over, we're sequencing it from the P5 region. Okay, and then once the sequencing is done, because remember, all our samples are mixed together, and those indexes are allowing us to group, to actually then demultiplex these samples and regroup them into specific samples that they belong to. And if you look at this image, each color represents a different index. So based on those specifics, the instrument will then decipher which reads belongs to which sample, and it will group those reads based on specific index to those samples. And this obviously happens on the instrument. All of this that I just spoke about happens on the instrument, and the instrument will convert those images to the base calls, and then it will do the demultiplexing of data, which in the end you will get your FASTQ files. Certain instruments can also do what we call a secondary analysis, where they do some kind of alignment or maybe even assembly, and they can also do some variant callings. But this can also be taken off the instrument and be done at the different pipelines. And also there is also interpretation of data, where we look at, what does this all mean, and what are the possible, let's say, treatments or preventions that we can do? What can we learn from this data that we just acquired? And just when it comes to sequencing, usually we talk about sequencing coverage and how much data do we need. Usually this is the question that always pops out. People ask how much data do I actually need? And obviously this will depend on how many samples you want to do. What is the output of that cartridge kit? How much you can actually do with the reagents that you have, and also how much depth, how much do you want to sequence? And when we talk about sequencing coverage, I always tell people, think about you stretching that DNA, and think about it as a road. Now that you stretch it, think about it as a road, and how many times do you need to walk up and down in order to cover certain regions, in order to make sure that that you picked up on that specific locus and what is the variation on that. And obviously more reads we have, yes, it increases the cost slightly, but that gives us increased sensitivity and confidence calling. So this is the power of NGS, because we can do a massive parallel sequencing, and we can do multiple targets at the same time. If we are not doing a whole genome sequencing, we can zoom in and we have so much data to look at, and we can then decide, is this maybe a bias, or is it maybe a sequencing error, or is it really a true variant when it comes to looking at things. So also Illumina has on their website what they call a sequencing coverage calculator, where you can actually say if you're working with DNA or RNA, and what is your different applications. Here you will see there are many different applications. You will just have to put in the size of your genome, and it will then help you decide what is the best instrument and how many samples you can multiplex on that specific instrument, but also on the specific different cartridges that are available, different kits that are available out there. But you're also more than welcome to reach out to us for this discussion. And then just to conclude, is that, of course, there are many bioinformatics solutions out there, and Illumina has a suite of these. When it comes to designing certain, let's call them panels, when it comes to, let's say, genetic disease research or oncology, where you want to look only at the targeted regions, on specific regions, you can use what we call a design studio to design such panel, unique custom panel for you. Then obviously we have on instruments we are able to do around setup and the secondary analysis, and also there is what we call a base, which is a cloud-based server out there, which has many different analysis pipeline for many different applications. And like I said, on the 14th of August, we're going to have a session just on analysis pipelines and what is possible. So with this slide, I would like to thank you for attending, and please do let us know if you have any questions. I just dropped out these two emails out there, but you're more than welcome to email any of us at separations.co.za, and we will work with you towards a successful NGS project. Thank you, everybody.